So good morning, everyone. Today we will discuss about the infertility concept. The infertility is termed as the inability of a couple. The infertility is termed as inability of a couple to conceive a child. To conceive a child. The inability, even after having unprotected coitus for almost all 2 years. Even after 2 years of unprotected coitus, still they are not capable of conceiving a child. So inability of a couple to conceive a child even after 2 years of unprotected coitus, that is termed as infertility. And there are particular reasons for the infertility. The reasons may be physical, psychological, and maybe because of some drugs or medicines. Many reasons. Next, immune system related to health issues. So there are many factors or reasons for the infertility. It may lead to infertility. There are many factors behind that. So how to overcome this infertility and to conceive a child? By overcoming and conceiving a child, how to do? So there are particular methods. These methods or technologies will be implemented to conceive a child. These technologies are applied here. So those technologies are called assisted reproductive technologies. Assisted reproductive technologies. Assisted reproductive technologies. Assisted reproductive technologies. They're nothing but arts. Assisted reproductive technologies. These technologies are implemented which helps the couple to produce a child. There are 5 different assisted reproductive technologies are there that we are going to study. The first one is IVF. Second one, GIFT, gift. And third one, ICSI. And fourth one, AI. And fifth one, surrogacy. These all are 5 methods of arts. Assisted reproductive technologies, 5 methods. 5 methods of assisted reproductive technologies. First, we'll start with IVF. IVF stands for in vitro fertilization. In, I for in, V for vitro, F for fertilization. So in vitro fertilization, IVF stands for in vitro fertilization. This program is also called test tube baby program. Test tube baby program. Test tube baby program. In vitro fertilization is also termed as test tube baby program. In this method, the female gamete from the mother is collected, semen from the father collected, sperms from the father collected. In the laboratory, in the test tube, this both gametes are allowed to fuse in a hygienic conditions. In a hygienic scientific conditions only, we are allowing the sperms and ovum, sperms and secondary oocyte to undergo fertilization process, and as a result, the zygote is going to be produced. Okay, and then the zygote we allow for the development of an embryo. From there, the embryo is collected and transferred to female individual. This female individual we are going to transfer. So the process I will write here. From the male, from the female, what are collected? Gametes. Here sperms are collected. Here secondary oocyte is collected. Secondary oocyte and test tube allow for fusion. Okay, allowed test tube fusion, and it results in the formation of zygote, and zygote we allow zygote to undergo cleavage process, and embryo is going to develop almost all morula stage. At morula stage, we collect this embryo. And then embryo is going to be transferred. That is called embryo transfer. Embryo transfer, or this embryo transfer is of two types. This embryo transfer into that female individual. This female individual we are transferring. Okay. Already gamete collected from the same female. The embryo which is produced in the laboratory is transferred to this female only. Thus, it will help in the growth of that embryo in the mother. Further growth. So the first embryo transfer is zygote intrafallopian transfer. Zygote intrafallopian transfer. The zygote or early embryo, both we have an option. Okay, zygote or early embryo. Zygote or early embryo. The zygote or early embryo is with up to 8 blastomeres. 8 blastomeres into fallopian tube, injected into fallopian tube. So that's why it is called fallopian transfer into fallopian tube. Next, second one. The second method is intrauterine transfer. Second method. Intrauterine transfer. That means the transfer of embryo with more than 8 blastomeres transferred into uterus directly. Okay. More than 8 blastomeres. The embryo with more than 8 blastomeres is transferred into uterus or injected into uterus. That's why intrauterine transfer it is called. Intrauterine transfer. So this is totally in vitro fertilization. This in vitro fertilization is also called test tube baby program. In this, from the male, sperms are collected, from the female, secondary oocyte is collected, and we allow for the fusion in the test tube in laboratory. See, vitreous means glass. Glass means test tube. Vitrio. Okay, in vitro. That means in the laboratory test tube conditions. In the test tube. The fusion in test tube. In vitro. Fertilization. It is also called test tube baby program. Male sperms, female secondary oocyte collected, allowed the fusion in test tube. Zygote produced. The zygote undergoes cleavage and embryo at morula stages or nearly before morula stage also can be done. The embryo transfer, that's why it is called. The embryo transfer into the female. First method is zygote intrafallopian transfer. Zygote intrafallopian transfer. This is commonly we call it ZIFT, ZIFT, and this is IUT, intrauterine transfer, IUT. But zygote intrafallopian transfer, early embryo only up to 8 blastomeres, only up to 8 blastomeres. Within up to 8 blastomeres, we transfer into fallopian tube of the female individual. We transfer. That is called zygote intrafallopian transfer. That's why, because transferring into fallopian tube, so that's why intrafallopian transfer we call it. And second method, intrauterine transfer. We collect the embryo with more than 8 blastomeres into the uterus. So that is called intrauterine transfers. All your doubts will be addressed at the end of the topic, please. Next. So this is about in vitro fertilization. In vitro fertilization. Now we are going to study the second method of assisted reproductive technology, that is GIFT. So hope you understood. Give thumbs up reaction if you have understood, so that I will start the next topic. GIFT, GIFT procedure. GIFT procedure I am going to continue. So GIFT stands for gamete intrafallopian transfer. Gamete intrafallopian transfer. It's very simple. Here one female will be acting as a donor, and from that female donor ovum is collected and transferred into the fallopian tube of the another female who cannot produce the ovum. So here one female is giving the gift to another female. We have to consider like that. So from the donor female, ovum is collected. And injected into fallopian tube of a recipient female who cannot produce ovum. So this method is called gamete intrafallopian transfer. But can provide suitable environment for fertilization. She can provide the environment for the fertilization, but she cannot produce ovum. So those kind of females, okay, but can provide suitable environment for the fertilization. And also implantation and development. Okay, so hope you understood this topic. Okay, now we are going to study the next topic, that is intracytoplasmic sperm injection. ICSI stands for I for intra, C for cytoplasmic, S for sperm, IFR injection. Intracytoplasmic sperm injection. Intracytoplasmic sperm injection. So it is a laboratory, completely laboratory procedure of fertilization, in which a single sperm from the male parent is injected directly into the egg or gamete of the female individual. So once again I am telling, the single sperm is collected. A single sperm is collected from the male parent. A single sperm and injected into the ovum of the female parent. Okay, from the male parent, single sperm is collected and injected into the ovum of a female parent. Okay, and after fertilization, okay, it is allowed for fertilization. After fertilization, this embryo is implanted into the woman's uterus. Embryo is implanted into the embryo's uterus. Female's uterus. Female's uterus. Implanted into the female's uterus. This method is called within the cytoplasmic. Here, ovum's cytoplasm of the ovum, we are injecting. Cytoplasm of the ovum, we are injecting sperm. Sperm we are injecting into the cytoplasm of the ovum. So cytoplasmic. Sperm we are injecting. So sperm injection. Intra means within the cytoplasm only we are injecting. No? So intracytoplasmic sperm injection. Very simple method. From the male parent, single sperm is collected and injected into the cytoplasm of the ovum. That's why it is called intracytoplasmic sperm injection. After fertilization process, that embryo is directly implanted into the female's uterus. So I think you have understood this method. Okay, the next method is artificial insemination method. Artificial insemination method. The insemination means we already know in the third chapter itself we studied what is insemination, right? The releasing of semen from the male copulating organ during the coitus into the vaginal tract, that is called insemination, right? First ejaculation, then followed by insemination, means releasing of semen into the vaginal tract. But here in this procedure, which is called artificial insemination, artificial, A for artificial, I for insemination, artificial insemination procedure. In this artificial insemination procedure, the semen collected from the male parent or husband or donor, two options we have, either male parent or any donor, we collect the semen. The semen is collected either from the male parent or donor, is artificially introduced, artificially introduced into the vaginal tract of the uterus of the female. Okay, introduced into the vaginal tract or uterus of the female, Okay. Suppose, sir, why to collect the semen from male parent? Yes, suppose the male parent is fertile only, naturally is fertile, but he cannot perform insemination. He couldn't perform insemination, so insemination is not happening during coitus, but he can produce semen. So those kind of parents, semen can be collected. Suppose he is infertile, from the donor they can collect this, okay, and artificially injected into the vaginal tract or uterus of the female. So this is called artificial insemination. Artificial insemination. And artificial insemination into uterus. Artificial insemination into the uterus is there, no? I have told. Vaginal tract is okay, artificial insemination. Then what about this artificial insemination into uterus? That is called intrauterine insemination. Intrauterine insemination. Intrauterine insemination. This is called IUI. Intrauterine insemination. If insemination is performed into the uterus, artificial insemination only, but into the uterus it is called intrauterine insemination. Intrauterine insemination. This technique is useful for the male partner having inability to inseminate. Inability to inseminate. Got it? Or some male parents who are having low sperm count. Low sperm count. The males with here. Number one, two conditions are there. First one, can't inseminate. The males who can't inseminate, and the other one, low sperm count. Low sperm count. Because if there is low sperm count, the possibility of fertilization will be very less. The male individual with the low sperm count, the fertilization possibilities are very less. So that's why the donor can be selected. Okay. Can't inseminate means that male parent can be selected. Semen is collected either from the male parent or donor. That's it. And artificially introduced into the vaginal tract or uterus of the female. This artificial insemination, if it is performing into the uterus, artificial insemination into uterus is called intrauterine insemination. That is IUI, intrauterine insemination. Okay. So hope you understood artificial insemination process. Artificial insemination process just now completed. And we are going to study the next topic. Now the next topic is surrogacy. Surrogate. Have you heard about surrogate mothers? Surrogate mothers, who is not actually a biological mother but carries embryo in her uterus. So they are called surrogate mothers. They are surrogate mothers. The surrogate mothers can bear a child for a couple. She can bear a child for couple. That mother is called surrogate mother. Okay, she can bear a child. Surrogate mothers are nothing but surrogate mothers. Surrogate mothers can bear a child. Can bear a child for the couple. For any couple. Okay. If the wife is infertile or unable to carry, whatever. Okay. If the female in this couple, if female is unable to carry, the female is unable to carry due to any reasons, right? Her uterus got damaged, or physiologically she cannot bear a baby child, or it may affect her like that. So female is when female is unable to carry, then the surrogate mothers can bear the child. This voluntarily, and they have to select properly. Okay, and the surrogate is impregnated. Surrogate mother is impregnated with a blastocyst. Okay, impregnated either by artificial insemination or implantation. By two methods we can do, right? Suppose semen collected from male parent or donor, no? Directly, instead of injecting into that female who is unable to carry, to directly surrogate mother only she can transfer. No? They can transfer the insemination, right? They can perform insemination. So either artificial insemination or the other method is Implantation directly, artificial implantation into the uterus directly. Implantation. Okay, it is direct implantation into the surrogate mother, or else the semen can be transferred to that surrogate mother's uterus or fallopian tube. Artificial insemination. That means how to transfer. You may get a question, no, sir, how to transfer it to surrogate mother? These are the two ways. Okay, we can transfer either semen or embryo directly. And coming to this, assisted reproductive technologies, either in vitro fertilization or gamete intrafallopian transfer or intracytoplasmic fermentation or artificial insemination or surrogacy. Whatever the method that you are going to select, it requires the high precision handling by a specialized professionals. Specialized professionals are required, and the handling and apparatus and procedures must be very, very careful. Okay, otherwise it will give you a lot of loss of money. Why? Because it is highly expensive methods, and instruments also highly expensive instruments. Therefore, these facilities are available only in a very few centers. Okay, because highly expensive instruments. You can find very less number of fertility centers. Fertility centers only these machines are present. And there are many factors which are associated with this assisted reproductive technologies. Okay, and coming to, instead of spending lots of lakhs of money, the infertility couple, if they are ready to spend lakhs of money for these assisted reproductive technologies, instead of that, they can adopt an orphan, right? Instead of spending a lot of money, they can adopt an orphan, right? It is equal to same, no, right? Anyhow, they want a child. There are many orphans in India, children. So legal adoption is a good method for a couple who are looking for these procedures. Instead of spending on these procedures, they can go for legal adoption. The adoption must be legal. As you see in the movies, going into some orphanage home, child orphanage home, and getting the kid in movies only. But in real life, you have to take a lot of procedure. You have to indulge in a lot of procedure. It's a big procedure in which you have to get the permission, and you will be evaluated, and who are your background, everything will be checked. Then only they will give permission to adopt a kid. It's not like what you see in the movies, going into orphan home or come to home. It is not like that. Okay, so there is a lot of big procedure is there for the legal adoption. So this is all about the assisted reproductive technologies. Now I'll just like to summarize, summarizing view I will write on the board for each method. Just 5 minutes. Within 5 minutes I'll finish it up. So artificial insemination, in vitro fertilization from the male, from the female, gamete, test tube, zygote, embryo transfer again, 2 types, embryo transfer 2 types, ZIFT, IUD, ZIFT and IUD. Very simple, no? Next, gift from the female donor. Okay, shortcut I will write. From the donor female gametes, fallopian tube of recipient female, over gift. Next, ICSI from the male or from the donor, 1 sperm injected into cytoplasm of the ovum, intracytoplasmic sperm injection, process over. Next, artificial insemination, either from the male parent or donor, semen is injected into fallopian tube vaginal tract, vaginal tract or uterus. If the artificial insemination done through uterus, that is called intrauterine insemination. Intrauterine insemination. Next, surrogacy. Surrogacy. Embryo or semen injected into, suppose if it is embryo impregnated into uterus, if it is semen injected into reproductive tract of the female, of surrogate mother, okay, uterus or vaginal tract of surrogate mother, For all 5 procedures, shortcuts for all 5 procedures I have written. Please note down. Once again, I'll tell if you want. From the male parent, gamete. From the female parent, gamete. In the test tube, fused, zygote produced, then embryo transfer, ZIFT, ZIUT. Zygote intrafallopian transfer, intrauterine transfer. If it is transferred through fallopian tube, less than 8 blastomeres, up to 8 blastomeres. Intrauterine transfer means more than 8 blastomeres. Over in vitro fertilization. And gift, the gamete intrafallopian transfer. From the donor female gamete is collected and transferred into fallopian tube of the recipient female. Over. Next, from the male or donor male, one sperm is from the male parent or donor male. One sperm is collected and injected into cytoplasm of the ovum. Next, artificial insemination. From the male or donor, semen is collected and transferred into vaginal tract or uterus. If this artificial insemination done through uterus, that is called intrauterine insemination. Surrogate mother. Into the surrogate mother uterus or vaginal tract, the embryo or semen is injected artificially. That's it. So this is all about the assisted reproductive technologies.